Hi, good morning. Happy to meet you all after a long gap. So, I am Dr. Jagdish. So, I'll be taking today's national test and discussion for biochemistry, and we'll be discussing important questions during this session. So, let's start the session. The first question is, during the peripheral nerve regeneration, which of the following motor proteins is primarily involved in the transport of materials along the axon? They are asking about which protein is responsible for transporting in an anterograde fashion. Okay, the options are dyneins, kinesins, myosin, and actin. Okay, the answer is kinesin. So these proteins are called as molecular motors. Okay, these proteins have ATPase and GTPase function, which are responsible for moving the organelles, proteins, and other cellular components within a cell or from a cell to cell. So what are the functions of these proteins? These proteins are involved in vesicular trans and myosins. Kinesin is a microtubule associated protein which is responsible for anterograde transport. That is, it will be responsible for moving the substances from the soma, that is cell body, to a presynaptic terminal. So this is soma. So this is presynaptic terminal. So kinesins will be responsible for moving the substances from the soma to the presynaptic terminal. So this is called anterograde transport, and this will be done by kinesins. The protein which is responsible for the reverse, that is from the presynaptic terminal to the soma, this is called retrograde transport, and this is done by dyneins. Okay, so with this, the answer for this question is this is kinesins which is responsible for anterograde transport. Okay, so that's about the first question. Okay, so just few information about these proteins. These proteins are responsible for various functions. Okay, they're mainly microtubule associated proteins. We already know about the third protein, myosin, which is responsible for muscle contraction. Okay, fine. So let's go to the next question. Enzyme. A 55-year-old male is diagnosed with methanol poisoning. Okay, and he is treated with another alcohol called ethanol. Okay, they already gave you the answer here, part of the answer here. They gave ethanol because it is a competitive inhibitor of an enzyme called alcohol dehydrogenase, thereby preventing the conversion of methanol to formaldehyde and formic acid, which are toxic. So which of the following enzyme kinetics will be observed by giving ethanol? Okay, so if you give a competitive inhibitor, what happens to Km and Vmax? That's the question. Okay, so the answer here is, if you introduce competitive inhibitor to a reaction, okay, it will increase the Km, but there won't be any change in Vmax. Okay, the answer is B. Okay, so what is Km? What is Vmax? Okay, Km is nothing, it's called as Michaelis-Menten's constant. Okay, so it's a Michaelis-Menten constant. Okay, so what is this? So Km is nothing but substrate concentration at which velocity of the reaction is equal to half the Vmax, half the maximum velocity. Okay, this is the Km. Okay, so Km is actually inversely proportional to affinity of the enzyme towards its substrate. So if an enzyme has high Km, then it has a low affinity towards that substrate. Okay, so this is about Km. Okay, then what is Vmax? Vmax is the maximal velocity of the reaction. Is the maximal velocity of the reaction. Okay, so these two things will change whenever an inhibitor is introduced into a reaction. Okay, so there are three types of inhibition. One is called competitive inhibition. Okay, The second one is called non-competitive inhibition. There's something called mixed, which is the features of both. Okay, so there are three types: competitive, non-competitive, there's something called mixed. Okay, let's see what is non-competitive inhibition first. Okay, here the inhibitor will bind either the free enzyme or enzyme substrate complex. What does that mean? So if you write an enzyme catalyzed reaction, this is how you can write. Okay, enzyme plus substrate forms enzyme substrate complex, which will be converted to enzyme and product. Okay, so the non-competitive inhibitor will bind either the free enzyme here or the enzyme substrate complex. Okay, so either the free enzyme or the enzyme substrate complex, and it binds to a site other than the active site. Okay, other than the active site in the enzyme. So when this happens, there won't be any change in the Km. For example, this is without the inhibitor. The blue line is without the inhibitor. See, there is no, this is the Km. When you add the non-competitive inhibitor, the Km remains the same, but the Vmax decreases. Okay, Vmax decreases. Okay, so that's about the non-competitive inhibition. Okay, what about competitive inhibition? Competitive inhibition, the inhibitor will be structurally similar to the substrate. Okay, so it is a structural analog. So the inhibitor and the substrate will compete with each other to bind to the active site of the enzyme. So when this happens, Km will increase. Okay, if you see here, without inhibitor, Km is here. But after you add the inhibitor, Km is pushed, that is increased to here. So Km will increase, but the Vmax will remain unchanged. Okay, so when you see a characteristic picture like this, it's a clue that there is a competitive inhibitor in the system. Okay, so that's about the competitive and non-competitive inhibition. So let's go back to this case. This case is an interesting case where patient is having alcohol poisoning because of methanol, and it is treated with ethanol because ethanol is a competitive inhibitor of alcohol dehydrogenase. So both will compete with each other, thereby the methanol will not be converted to formaldehyde, which is more toxic to the system. Okay, that's it. Okay, so that's about the inhibitors. Okay, fine. Now let's go to the third question. 3-week-old exclusively breastfed male neonate presents to pediatric clinic with history of following symptoms: frequent foul smelling and frothy stools. Okay, the mother reports that infant has been increasingly irritable after feeding and exhibited episodes of unexplained crying and arching of the back. There is no history of recent antibiotic. There is no history of formula fits. Okay, so what is this about? Which of the following tests can be performed to evaluate the present condition in this patient? Okay, so presentation itself giving you a clue. Okay, frequent foul smelling, frothy stools after breastfeeding. No history of antibiotics, no history of any other formula fits. Okay, this is a clear-cut case, or this is an indication of a condition called lactose intolerance. Okay, so this baby is more likely to have lactose intolerance. The diagnosis is lactose intolerance. Now the question is, which of the following test is more appropriate to evaluate this present condition, to evaluate lactose intolerance? Okay, they have given you 4 options. Let's see one by one. Hydrogen breath test, stool reducing serum lactose level, and endoscopy with biopsy. Okay. See, before going to the options, we have to know what is lactose intolerance and what is the cause of, what is the biochemical basis for these symptoms seen in lactose intolerance. Okay. See, lactose intolerance is caused due to a deficiency of an enzyme called lactase. Okay, so lactase is a disaccharidase. It acts on lactose. Okay, lactose is a disaccharide which is made up of two monosaccharides, glucose and galactose. Okay, so normally lactase, what it will do is it will convert lactose to form glucose and galactose. This is the normal function of lactase, and this is present in the intestinal brush border along with other disaccharidases like sucrase, maltase. Okay, sucrase isomaltase complex, so on so. Okay, it is present in the intestinal brush border. Okay, if somebody is not having, if there is deficiency of lactase, lactose cannot be metabolized. Okay, so this step will not happen. Then lactose will accumulate. Okay, so what? Okay, the problem starts because of this. Okay, so the lactose accumulates in the gut. This is the intestine. Okay, lactose accumulates in the intestine. Okay, so because of lactase deficiency, lactose accumulates. Okay, what happens to this lactose is lactose will be acted upon by bacteria, because intestine contains a lot of bacterial microorganisms. Your bacteria, intestinal flora contains bacteria. Okay, so this bacteria will act on this lactose and convert them into lactic acid, 1, carbon dioxide, hydrogen, sometimes methane also. Okay, fine. Then this lactose is osmotically active. So what happens? Water molecules present in the blood or present in the interstitium, present in the capillaries, will enter into the intestine. So there will be a lot of water in the intestine. Okay, so this will cause diarrhea, and because of this because of this gas production and methane production, there will be flatulence. 1. There will also be abdominal pain. They will also have bloating. Okay, so this is why patients with lactose intolerance present with diarrhea, abdominal pain, bloating, and flatulence. Okay, so because of this diarrhea, they will have electrolyte abnormalities. Okay, fine. Now let's go back to the investigations of choice. Okay, now if you go back to the choice investigation of they have listed out, they have given 4 investigations: hydrogen breath test, stool reducing substance, serum lactose, endoscopy with biopsy. Okay, let's go to the hydrogen breath test. Yes, you can do hydrogen breath test to diagnose lactose intolerance, because when the lactose undergoes fermentation with bacteria, it releases hydrogen peroxide, which will eventually come out in the breath. So this can be tested by hydrogen breath test. Yes, hydrogen breath test is a test for lactose intolerance, but it is done only in older children and adults, not in newborns. Okay. Then what is the test for newborns? Newborns, the test is stool reducing substances. Okay. Collect a stool sample from the baby and check for reducing substances in the stool. Okay, so what are the reducing substances you will find? You will find more likely the lactose, because lactose is not digested. Lactose will come out in the stool, which will be answered by this test. So the test which you will do in this patient is stool reducing substances. So the answer for this question is stool. So can we measure serum lactose? No, it won't be any useful. It won't be of any use, because serum lactose levels will not change that much with patients with lactose intolerance. So this test is not done. Endoscopy with biopsy, yes, you can do endoscopy with biopsy, but it's an invasive procedure. Okay, it is done only at the last option. Okay, not for the initial evaluation of suspected lactose intolerance, especially in neonates. Okay, so out of these 4 tests, you can do 3 tests. Okay, hydrogen breath in children and adults. Stool reducing substances, especially in the neonates. Serum lactose levels, no, this test cannot be done. This is of no use. Okay, endoscopy with biopsy, yes, it is invasive, but it is not done as the early evaluation. Okay, so that's about lactose intolerance. Okay, fine. One more important point about this lactose intolerance is that lactose intolerance can be congenital. Okay, so you can see patients with lactose intolerance right from birth. Okay, so it can be congenital, which is more likely to be the cause in this patient. Okay, second, it can be age induced. Okay, that is the lactase expression in the gut decreases with age. Decreases with age. Okay, so elderly patients are more likely to have lactose intolerance. Okay, fine. So that's about this. Okay, fine. So how do you treat this? We can give lactase supplements. Okay, sometimes formula feeds also work. Okay, so that's about it. Okay, fine. Now let's go to the next question. Okay, so that's about this question. Now let's go to the next question. Okay, fine. Okay, 24-year-old female presents to emergency department with symptoms of severe hypoxia despite adequate oxygen supplementation. She has a history of a condition called hereditary spherocytosis. It is a condition known to affect the red blood cells. Okay, they have done some laboratory tests and they found out that in the red blood cells, the concentration of 2,3-BPG is very high. The red cell RBC 2,3-BPG is high. So, which of the following enzymatic steps in the glycolysis is bypassed by the pathway that leads to the accumulation of 2,3-BPG in her red blood cells? Okay, so that is a question. Okay, basically they are asking this question to test your knowledge on a pathway called Rappaport Leu-Behring shunt. Okay, which is a adaptation of, or which is a shunt pathway of glycolysis found exclusively in the RBC. Okay, fine. So let's see what is that pathway. Okay, see, normally glycolysis happens like this. Glucose is converted to glucose 6-phosphate where one ATP is used. Okay, then glucose is converted to fructose 6-phosphate by an isomerase step. Then this is converted to fructose 1,6-bisphosphate. Okay, now this will split into two to form glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, both are isomers. Okay, this glyceraldehyde 3-phosphate will be converted to 1,3-bisphosphoglycerate. Okay, here one NADH is released. Okay, and this 1,3 will be converted to 3-phosphoglycerate. Here one ATP will be released. This 3-phosphoglycerate will be converted to phosphoenol pyruvate, and this phosphoenol pyruvate will be converted to pyruvate. Here again one ATP is released. Okay, so this is the normal classical glycolysis step. Okay, so what happens is this glycolysis will undergo certain modification in RBC. So what is the modification that you will see in the RBC here? In this step, this 1,3-bisphosphoglycerate will be converted to 2,3-bisphosphoglycerate. Okay, by an enzyme called phosphoglycerate mutase. Okay, and this will be converted to 3-phosphoglycerate by an enzyme called 2,3-bisphosphoglycerate phosphatase. So one phosphate will be released here. Okay, fine. So this is what happens in RBC. Okay, this is a shunt pathway that happens in RBC. This name of this pathway is, this shunt pathway is called Rappaport-Lubering shunt, or also called as RL shunt. Okay, so why this happens in RBC? So why RBC has to produce this 2,3-BPG? Okay, that's because of two reasons. Which is because of main reason that 2,3-BPG. Okay, is responsible for modulating the affinity of oxygen towards hemoglobin. Whenever 2,3-BPG binds to hemoglobin, it decreases the affinity of hemoglobin towards oxygen. So it favors unloading of oxygen. So it favors delivery of oxygen. This happens especially in people living with high altitudes or when there is hypoxia. So whenever there is hypoxia, 2,3-BPG levels in the RBC will increase. Okay, so this is the background which you should know before you approach this question. Okay, now let's go back to the question. Okay, so this is an alternative. Question is, which of the following enzymatic steps in glycolysis is bypassed by this shunt pathway and leads to accumulation of 2,3-BPG in the red blood cells? Okay, the answer is this. The phosphoglycerate kinase step here is bypassed. So this step is bypassed. Okay, so that's the reason. So this is the phosphoglycerate kinase step. This step is bypassed in Rappaport-Lubering shunt. Okay, fine. So that's about this. The answer is phosphoglycerate kinase. Okay, rest of the steps will be normal. Everything will be normal. Only in this step, 2,3-BPG, it enters into a shunt pathway and come back to the same glycolytic intermediate. So this pathway is bypassed in Rappaport-Lubering shunt. Okay. So instead of 3-phosphoglycerate, 1,3-bisphosphoglycerate is converted to 2,3-bisphosphoglycerate. Okay. So this thereby it prevents one ATP generation at this point. Okay. So that's about this pathway. Okay. I told you the physiological significance of this pathway because 2,3-BPG decreases the affinity of oxygen towards hemoglobin so that hemoglobin can deliver the oxygen efficiently, and this will happen only during hypoxia. Okay. But why this patient is having this condition? Because of this hereditary spherocytosis. So what is hereditary spherocytosis? Hereditary spherocytosis is actually a condition caused due to a defect in a RBC membrane protein called spectrin. So what is the function of this protein? This protein is responsible for linking RBC inner membrane to outer layer. So because of this defect, the RBC membrane integrity is lost. Integrity is compromised or lost, leading to membrane damage and hemolysis. So because of this, there will be hypoxia. So even if you give oxygen, it may not be able to correct it. Okay. So there is hypoxia. So the red blood cells will automatically increase the concentration of 2,3-BPG. So that's the explanation why there is increased 2,3-BPG in this patient. Okay, fine. So that's about this question. Okay. One more modification that happens to RBC, that happens to glycolysis in the RBC is RBC doesn't have any mitochondria. Great. So what happens is, whatever pyruvate that is produced will not enter mitochondria for TCA cycle and then electron transport chain and oxidative phosphorylation to produce ATP. What happens to this pyruvate is everything will be converted to lactate by an enzyme called lactate dehydrogenase. Okay, this is nothing but kind of anaerobic glycolysis. Okay, but here oxygen is there, but mitochondria is not there. So pyruvate will not be converted to acetyl CoA. Instead, pyruvate will be converted to lactate. Okay, this is one more modifications that will happen in the RBC, especially for glycolysis. Okay, fine. Now the next question, question number 5. A 25-year-old athlete visits sports medicine clinic complaining of excessive fatigue and cramps during high intensity training. The physician explains the biochemical pathways that regulate the energy production in the muscle cells during the exercise. He highlights the role of oxygen availability and certain key enzymes in the metabolic processes involved. So, which of the following statements best describe an effect called Pasteur effect in the context of cellular metabolism, particularly relating to the role of an enzyme called phosphofructokinase? So they are asking you this question to check the knowledge of what is Pasteur effect, okay, and what is its connection with an enzyme called phosphofructokinase. Okay, now let's see what is Pasteur effect first. Okay, Pasteur effect is actually so when it comes to carbohydrate metabolism, there are few effects or hypotheses you should know. One is Pasteur effect, the other one is, I will tell you one by one. Okay, Pasteur effect. What is Pasteur effect is? Pasteur effect is inhibition of glycolysis in the presence of oxygen. It is basically found in yeast cells. Okay, so whenever there is increased levels of oxygen, glycolysis is inhibited, so that glucose is not fermented to alcohol. There is increase in glycolysis or fermentation. Okay, this is one. Okay, so everything will be, the energy for the cell will be derived from TCA cycle, electron transport chain, and so on. Okay, this is Pasteur effect. How this happens? Because oxygen inhibits an enzyme of glycolysis called phosphofructokinase. Okay, which is the rate limiting enzyme of glycolysis. So when this enzyme is inhibited, glycolysis is inhibited, phosphofructokinase 1. Okay, what's the function of this enzyme? This is the enzyme which will convert fructose 6-phosphate to form fructose 1,6-bisphosphate. So this step is inhibited in the presence of oxygen. So that's the reason why in the presence of oxygen, glucose is not undergoing glycolysis or glucose is not undergoing fermentation to form alcohol in the yeast. So this is observed by Pasteur and this is called Pasteur effect. Okay, so now let's go to the question. The question is, which of the following statements best describe the Pasteur effect in the context of cellular metabolism, particularly relating to the role of phosphofructokinase? Okay, let's see the first option. Pasteur effect enhances glycolysis in the presence of high oxygen. No, that's wrong. Pasteur effect inhibits glycolysis. Yes, first part of the sentence is correct. In the presence of oxygen, correct. By promoting the activity of phosphofructokinase, that is wrong. Okay, if phosphofructokinase activity is promoted, glycolysis will happen more. Glycolysis will not get inhibited, so that is wrong. Okay, Pasteur effect describes a shift from aerobic to anaerobic metabolism during intense exercise, resulting in increased phosphofructokinase activity. Again, that is also wrong. Okay, Pasteur effect inhibits fermentation in the presence of oxygen, correct, resulting in the inhibition of phosphofructokinase activity. That is correct. Okay, the answer is D. Okay, so that's what I told you. In the presence of oxygen, this phosphofructokinase is inhibited When this is inhibited, fermentation or glycolysis is inhibited. That's it. Okay, the answer is D. Okay, so in addition to this, you have to know two more effects. Okay, one is Pasteur effect. There's one more effect called Warburg effect. Okay, Warburg effect is a metabolic adaptation found in the cancer cells. This is a very important point you should know. Metabolic adaptation found in cancer cells or observed in cancer cells. What happens here? See, in cancer cells, it has been found that the mitochondria is not normal. Mitochondria is little bit is damaged. So what happens is, even in the presence of oxygen in the cancer cell, even in the presence of oxygen, cells will use or cells will utilize glucose for ATP by anaerobic glycolysis. Okay, cells will use glucose for ATP by anaerobic glycolysis. That is persistent anaerobic glycolysis even in the presence of oxygen. Okay, that is called Warburg effect. So it's a metabolic adaptation observed by the cancer cells, because glycolysis itself can give you some ATPs. Without mitochondria, glycolysis can produce 2 ATPs. Okay, so this will happen in a rapid fashion, so the cancer cells will be able to survive for a long time. Okay, and this will be enough to support their uncontrolled proliferation. So this is observed by Warburg, and this is named after him as Warburg effect. Okay, persistent anaerobic glycolysis even in the presence of oxygen, present mainly in the cancer cells. Okay, this is Warburg effect. Okay, there's one more effect which is slightly difficult to understand, but let me put it forward. It is called Crabtree effect. Okay, so what is Crabtree effect? Crabtree effect is high glucose will inhibit glycolysis. High glucose will inhibit respiration. That is, high glucose will inhibit respiration. So whatever glucose that comes inside will be utilized to undergo fermentation to form ethanol. This is also observed in yeast. Okay, so this is the effect of glucose on suppressing respiration in the yeast. That is Crabtree effect. Okay, fine. Now I have seen three effects. Okay, Pasteur effect, Warburg effect, Crabtree effect. Okay, fine. So that's about this three effects. Okay, so let's go to the next question. Which of the following inhibitor of TCA cycle specifically interferes with iron containing enzymes, leading to disruption of cycle's function? Okay, so here they are saying that there's an inhibitor which specifically acts on an iron containing enzyme. Okay, so what is that inhibitor? Okay, so the answer is fluoroacetate. Okay, first we have to know what are all the iron containing enzymes present in mitochondria, because TCA cycle happens in mitochondria. Okay, iron containing enzymes present in mitochondria. Okay, mitochondria is very important organelle because it is a powerhouse of the cell. Okay, because TCA cycle happens in mitochondria, electron transport chain and oxidative phosphorylation happens in mitochondria, both of which are essential for producing ATPs. Okay, so what are the iron containing enzymes present in the mitochondria? Okay, so these are the enzymes. First one is aconitase, which is a part of TCA cycle. Second, NADH dehydrogenase is a part of electron transport chain complex 1. Succinate dehydrogenase complex 2.
This is also part of TCA cycle. Cytochrome C oxidoreductase complex 3, which is also part of electron transport chain. So these are the 4 enzymes which contains iron in them, okay, particularly as iron sulfur clusters, okay. So among this, which enzyme is part of TCA? The answer is aconitase. So the answer for this is, what is the inhibitor that inhibits aconitase? So that's the question, okay. So the answer is fluoroacetate, okay. So this is a normal TCA cycle, okay. Oxaloacetate combines with acetyl-CoA, which is derived from pyruvate. The name of this enzyme is called pyruvate dehydrogenase, okay. And this acetyl-CoA combines with oxaloacetate to form citrate. Citrate is acted upon by this enzyme called aconitase to form isocitrate. Isocitrate will be acted upon by isocitrate dehydrogenase to form alpha ketoglutarate. One NADH is produced. This is acted upon by alpha ketoglutarate dehydrogenase to form NADH, to form succinyl-CoA and NADH. Succinyl-CoA is acted upon by thiokinase to form succinate. Succinate is acted upon by succinate dehydrogenase. Again, this is also an iron sulfur cluster containing protein. This is also an iron sulfur cluster containing protein, okay. To form fumarate, fumarate is converted to malate, malate is converted to oxaloacetate. So this is TCA cycle, okay. So here this aconitase is inhibited by fluoroacetate. That is the name of the inhibitor. So how this inhibits? Okay, there's something special about this inhibitor. What is that? Is this fluoroacetate combines with this acetyl-CoA to form fluoroacetyl-CoA. First, this fluoroacetyl-CoA combines with oxaloacetate to form fluorocitrate. Now fluorocitrate and citrate are somewhat similar, so they'll inhibit aconitase. So that citrate will, so that is how fluoroacetate inhibits aconitase. Okay, fine. Now citrate accumulates. Citrate is an allosteric inhibitor of phosphofructokinase, which is the rate limiting enzyme of glycolysis. So glycolysis is inhibited. One. Okay. Then since there is isocitrate is not produced, alpha ketoglutarate is not produced. Alpha ketoglutarate is responsible for producing glutamate, which is responsible for fixing ammonia. So ammonia will increase. Okay. So if fluoroacetate is used, okay, it will inhibit glycolysis, so ATP is not produced. At the same time, it can increase the ammonia levels, leading to hyperammonemia. Okay. So these are the additional points you should know. Okay, fine. So a little bit about aconitase. There's something special about aconitase. Aconitase is also called as moonlighting enzyme. This can be asked as an MCQ. Okay, moonlighting enzyme. Why it is called moonlighting enzyme? Because it has two different functions. Okay, what is it? So aconitase exists is involved in TCA cycle. So it exists in mitochondria, where it converts citrate to isocitrate. Okay, aconitase is also found in the cytoplasm. Okay, where it also converts citrate to isocitrate. Okay, isocitrate. But here, what will happen is, and this isocitrate will be acted upon by isocitrate dehydrogenase, which is also found in the cytoplasm, to form alpha ketoglutarate. Here it will produce NADPH. The cytosolic isocitrate dehydrogenase produces NADPH, whereas mitochondrial isocitrate dehydrogenase will produce NADH. Okay, same will be produced, alpha ketoglutarate. Okay, one. Second, this aconitase acts as an iron regulatory protein. Okay, so whenever iron levels are high in a cell, okay, what happens? So there are some iron regulatory proteins. So this iron will go and bind to this iron regulatory protein. And this iron regulatory protein can go and bind to certain mRNAs, okay, which contains, these iron regulatory proteins can go and bind to certain mRNAs. Okay, so these mRNAs will have like this. Okay, this could be 5' in, this could be 3' in. Okay, and there will be some, these are called iron regulatory elements. Iron regulatory elements. So what happens is this iron regulatory proteins can come and bind to this. Okay, and can either prevent the translation of certain proteins or promote the translation of some proteins, okay, based upon the location of these iron regulatory elements. Okay, some of the iron regulatory elements for certain proteins are present in the 3' end. Okay, so aconitis has this function. Okay, aconitis can act as an iron regulatory protein and can promote the translation of certain proteins or vice versa, depending upon the iron availability in the cells. Okay, so iron regulatory proteins are excellent regulators of cellular iron homeostasis. So aconitis is one such protein. Okay, aconitis has an enzymatic function in citric acid cycle in mitochondria. It also exists in cytoplasm. Okay, that is responsible for producing NADPH. Okay, which can be used for fatty acid synthesis in the cytoplasm. Okay, again, aconitis has an additional function as iron regulatory protein. Okay, so that's about this aconitis. That's why aconitis is also called as a moonlighting enzyme. Okay, it has diverse functions. Okay, fine. Now let's see about the other enzymes. Okay, of TCA cycle. Yes, there is something called citrate synthesis, which is not an iron sulfur cluster. Acetyl-CoA, yes, it produces pyruvate dehydrogenase. Okay. NADH can inhibit citrate synthase. Malonate can inhibit succinate dehydrogenase. Very important point. Okay, so malonate is a competitive inhibitor of complex 2. Again, succinate dehydrogenase is an iron sulfur cluster protein. Fluoroacetate inhibits aconitase, we just now saw. It is an iron sulfur cluster protein. Arsenite can inhibit alpha ketoglutarate dehydrogenase. So these are the various inhibitors of electron transport, TCA cycle. So alpha ketoglutarate is inhibited by arsenite. Okay. And then citrate synthase. Here it can be inhibited by citrate. It can also be inhibited by NADH. NADH. Then succinate dehydrogenase is inhibited by malonate. Okay. Then this acetyl-CoA can inhibit pyruvate dehydrogenase feedback inhibition. That's it. Okay. So these are the various inhibitors of TCA cycle. Okay. And there are many enzymes which contains iron sulfur in their protein, in their structure. Okay. So the next question is 45-year-old male with history of chronic alcoholism presents with increased oxidative stress and liver dysfunction. Okay. Laboratory tests reveal impaired detoxification of drugs and increased levels of reactive oxygen species. Okay, fine. Which of the following biochemical pathways is essential for preventing oxidative damage? Okay, so the answer here is HMP shunt. HMP shunt, two important functions of HMP shunt. One, it produces ribose 5-phosphate. Second, it produces NADPH. Ribose 5-phosphate is used for building up of DNA and RNA. NADPH has very important functions. Okay, one, synthesis of fatty acids, fatty acid synthesis, steroid synthesis, then antioxidant. I'll tell you how. Okay, so these are the various important functions of NADPH. Okay, fine. See, in HMP shunt, is mainly responsible for maintaining the integrity of RBC membrane. So what is the connection? RBC membrane integrity. So what is the connection? So RBC is a cell which is constantly involved in transportation of oxygen and carbon dioxide. Okay, and this oxygen has to be delivered in the tissues, and carbon dioxide should be picked up from the tissues and to be exhaled out. Okay, because of this constant transport of oxygen, RBCs, in the RBCs, what happens if you draw an RBC like this? Okay, so in the RBC, what happens is there are a lot of oxygen. Okay, when this oxygen gets partially reduced, it forms hydrogen peroxide, which is very high in. Okay, and this hydrogen peroxide is actually a reactive oxygen species. So it can damage the DNA, it can damage the membrane, leading to hemolysis. 1. Okay, so here what happens is, but RBC has a system in place to reduce this hydrogen peroxide to water. Okay, how that is done? That is done by this enzyme called glutathione peroxidase. Okay, so glutathione peroxidase will convert this hydrogen peroxide to form water. During this process, glutathione will get reduced. So glutathione will donate its 2 hydrogen atoms and becomes oxidase like this. What happens is, now we have to replenish this glutathione back to its reduced state. So this is a reduced state. This is something called oxidized state. Now we have to reduce this glutathione back, for which we need NADPH. And this NADPH comes from HMP shunt. Okay, so this NADPH will convert this glutathione back to reduced glutathione, so that this is ready for the next step of detoxifying hydrogen peroxide. So when this HMP shunt is not functioning, for example, there's one enzyme which is part of HMP shunt, which is called glucose 6-phosphate dehydrogenase, famously called as G6PD. So when G6PD is deficient, what happens? NADPH cannot be produced. Okay, so NADPH levels will come down. When NADPH levels come down, so what happens? This step is blocked. So everything will be glutathione. There is no reduced levels of glutathione, so glutathione levels will come down. So hydrogen peroxide levels will go up. Okay, that causes hemolysis. So there will be some hemolysis. That's why G6PD deficiency causes hemolysis. Okay, like this, NADPH is necessary for antioxidant action in various cells. I'm just giving you one example. Okay, so coming back to this case here, in this patient, he is a chronic alcoholic. Okay, so whenever somebody is chronic alcoholic, okay, so alcoholism can cause liver damage. 1. Okay, second, whenever the liver is damaged, the metabolism of drugs then the systems in place to get rid of reactive oxygen species will be compromised. Okay, so they will end up in oxidative stress. Okay, fine. So that's the reason why this patient need a lot of NADPH to prevent this reactive oxygen species induced damage, which comes from HMP shunt, which is one of the source for NADPH. There are other sources for NADPH also. For example, malic enzyme is also a source for NADPH. We just now saw one enzyme, cytosolic isocitrate dehydrogenase. It's also a source of NADPH. Okay, fine. So that's the reason. Okay, fine. So that's the answer for HMP shunt. Okay. One few more points about this G6PD induced hemolysis. Okay, This, whenever G6PD is deficient, okay, there will be hemolytic anemia. Okay, so G6PD deficiency causes hemolytic anemia. So there are certain triggers for this. That is, the patients might have G6PD deficiency, but they don't exhibit hemolysis all the time. So there are certain triggering factors for hemolysis in those with G6PD deficiency. What are those? Infections, fever, intake of drugs like antimalarials, and even some antibiotics can trigger hemolysis. 1, 2, 3. Then there's something food intakes. For example, there's something called fava beans. When fava bean intake is high, that can also trigger hemolysis. So these are some additional points with G6PD deficiency. And G6PD deficiency is most commonly seen in Africa. Okay, where malaria is also common. So what happens is, it is like a survival advantages to Africans to escape malaria, because malaria parasite needs intact RBC. Okay, so this is one of the survival advantage that is conferred by this mutation, which is found mostly in the African people. Okay, especially against Plasmodium falciparum. Okay, and so these are some additional points with the G6PD deficiency. Okay, fine. Okay, let's go to the next question. A 7-year-old boy presents with frequent episodes of fasting hypoglycemia, fatigue, and muscle cramps during exercise. His growth is delayed. Lab investigations reveal markedly reduced blood glucose levels during fasting. So there is fasting hypoglycemia. And when they did a when they did a liver biopsy, they found no glycogen in it. So glycogen is absent in the liver biopsy. Glycogen granules are usually seen in liver biopsy because glycogen is stored in the liver. So there is no glycogen in the liver. So which enzyme is likely defective? The answer is very clear here. No glycogen. So glycogen is not synthesized. The answer is glycogen synthesis is defective. Okay. So this is one of the glycogen. This comes under glycogen storage disease. Okay. So glycogen synthesis deficient here. There are various glycogen storage disease. Okay. They are from type 1 to type 7. Okay. Type 1 is called von Gioch's disease, caused due to defect in an enzyme called glucose 6-phosphatase. Okay. See, glycogen is a storage form of glucose. It's a branched form of glucose stored mainly in two areas: liver and skeletal muscle. Okay, liver and skeletal muscle. And then this glycogen will be break down to form glucose when our body needs it. So when this will happen? During fasting, during exercise, you need more glucose. So glucose comes from two sources. One, it comes from glycogen breakdown. This is called glycogenolysis. After this has been done, then body will look for glucose by a process called gluconeogenesis, which is synthesis of glucose from non-carbohydrate precursors. Okay, so this is glycogenolysis. When there is a defect in the breakdown of glycogen to form glucose, when there is a defect in this glycogenolysis, this predominantly leads to a condition called glycogen storage disease. Okay, so there are various enzymes involved in this. Okay, so first enzyme is glycogen phosphorylase. I am telling you about glycogen breakdown. That is glycogenolysis. What are the enzymes I am telling you? First enzyme is glycogen phosphorylase, responsible for the first step. There's something called debranching enzyme. That's the second enzyme. As I told you, glycogen breakdown can happen in both liver and muscle. So glycogen phosphorylase exists in muscle, exists in liver. Then there's something called debranching enzyme. Okay. Then there's something called acid maltase. Okay, there's one enzyme called acid maltase, which is not part of glycogen breakdown, but it is present in lysosomes. Okay, so when any of these enzymes are defective in glycogen breakdown, that can cause glycogen storage disease. Okay, and there are one more enzyme which is called glycogen synthase, which is responsible for producing glycogen. Okay, so that is also, when it is defective, can cause glycogen storage disease. Okay, and we are seeing one enzyme repeatedly from previous questions. One is called phosphofructokinase, which is an enzyme of glycolysis. When that is defective, that can also lead to glycogen storage disease. Okay, so these are the 5 enzymes. Okay, actually 6: muscle glycogen phosphorylase, liver glycogen phosphorylase, debranching enzyme. There's something called acid maltase, which is present in lysosomes, responsible for glycogen breakdown in the lysosomes, but the physiological significance of this enzyme is not known. Okay, but you should remember this enzyme called acid maltase. Then there's a glycogen synthesizing enzyme called glycogen synthase, and there's phosphofructokinase, and there's one more enzyme called branching enzyme, like debranching enzyme, which is responsible for breaking down glycogen. There's one enzyme called branching enzyme, which is responsible for producing glycogen. That is called branching enzyme. Okay, so these are the various enzymes. When they are defective, can lead to glycogen storage disease. Let's see one by one here. Okay, see, von Jerk's disease is caused due to an enzyme called glucose 6-phosphatase. Okay, this enzyme I have not listed out in the previous, when I listed out the enzymes, which you should remember, because glycogen, when it gets broken down, okay, it contains n number of carbon atoms. Okay, we don't know how many number of carbon atoms. So what is the enzyme I told you? This is broken down an enzyme called glycogen phosphorylase, which can be present in muscle or liver. When this enzyme acts on glycogen, what happens is it releases one glucose as glucose 1-phosphate. Okay, and what happens is so one glucose is removed from this glycogen, so glycogen will become N-1. Now this glucose will be taken to liver. Okay, where it will be converted to glucose 6-phosphate. So if your body needs glucose, this glucose 6-phosphate will be acted upon by an enzyme called glucose 6-phosphatase, which is present only in the liver, to form glucose. So that's how glycogen is broken down to form glucose. So glucose 6-phosphatase is also an enzyme for glycogen breakdown. Okay, when this enzyme is defective, it leads to a condition called type 1 glycogen storage disease, or called von Jerk's disease. Okay, so this is a very important question that been asked repeatedly in the previous year MCQs. Okay, von Jerk's disease is caused due to an enzyme called glucose 6-phosphatase. It's a glycogen storage disease. Okay, next comes this acid maltase, responsible for breaking down glycogen in the lysosomes. Okay, so when this enzyme is defective, it leads to a condition called Pompe's disease. Okay, so we'll take a small break here. We'll start again with this slide.